So, good day everyone. So, we are going to report the hydrogen bonds in protein and carbohydrates.
Hydrogen bonding is the interaction involving a hydrogen atom located in a pair of other atoms having a high affinity for electrons. It can exist between atoms in different molecules or in parts of the same molecule. Hydrogen bonds are also responsible for holding together DNA, proteins, and other macromolecules. And also, hydrogen is a special type of dipole-dipole attraction between molecules. It results from the attractive force between a hydrogen atom covalently bonded to a very electronegative atom, such as nitrogen, oxygen, or fluorine atom, another very electronegative atom.
So, hydrogen bonding in proteins. Proteins are large-sized molecules, or also called as macromolecules, polymers of structural units called amino acids. Amino acids can be released from proteins by hydrolysis. Proteins are chains of amino acids that are connected by peptide bonds and folded into three-dimensional structure. The side chains of the individual amino acid residues determine the interactions among amino acid residues and ultimately the folding of the protein. They do most of the work in cells and are required for the structure, function, and regulation of the body tissues and organs. Is a highly complex substance that is present in all living organisms, and proteins are of great nutritional value and are directly involved in the chemical processes essential for life. So, yung naka-underline na word dyan is yung hydrolysis. Meaning yan is it is a chemical breakdown of molecule due to the reaction with water. Next is yung type naman ng proteins. First is yung antibodies. These are specialized proteins that has a function to defend and protect the body against antigens or foreign invaders. Their ability to travel through the bloodstream enables them to be utilized by immune system to identify and defend against bacteria, viruses, and other foreign intruders in blood. So, ito lang yung parang mga white blood cells na nagpo-protect sa katawan natin sa mga bacteria and other viruses. Next is yung contractile proteins. These are responsible for muscle contraction and movement. Examples naman ito is yung actin and myosin. Pag enzymes naman, these are proteins that facilitate and speed up biochemical reactions, which is why they are often referred to as catalysts. Yung mga protein enzymes is responsible for the metabolism and catabolism sa body. Hormonal proteins are messenger proteins that help coordinate certain bodily functions, and examples nito is yung insulin, oxytocin, and somatotropin. Next is yung structural proteins from the word itself, structure. These are fibrous and skinny, this formation making them ideal for supporting various other proteins such as keratin, collagen, and elastin. Storage proteins naman, from the word itself, storage, this reserves amino acids for the body until it is ready to use. Last is the transport proteins. These are carrier proteins that move molecules from one place to another in the body. Next naman is yung 4 levels of structural organization ng globular proteins. Yung amino acid sequence or yung primary structure is responsible for the higher level structure and biological function of a protein. So, ito yung primary structure niya. As you can see, yung mga bilog-bilog na maliliit na yan, ang tawag dyan is amino acid, and yung nagko-connect na parang tale or yung black strand is called, yan yung tinatawag nila na peptide bond. Next is yung the secondary structure that defines the conformation of the polypeptide backbone with the typical repetitive elements, alpha helix and beta pleated sheet. Ito naman siya. Yan, yung Tinatawag nilang side chains dyan is yan mismo, itong structure na to. And ayun nga, yung mga maliliit na to is called amino acids. And next naman, the arrangement in three-dimensional space of the regions with secondary structure elements and the regions of irregular polypeptide conformation is called tertiary structure. Yung nakikita niyo sa screen, yung tertiary structure with that involved na yung hydrogen bond and yung local folding ng polypeptide chain is present sa beta sheets. Next is yung quaternary structure. It describes the folding of the polypeptide chains into functionally active sites and is, above all, essential for the specific biological properties of a protein. Ayan, ito naman siya, yung quaternary structure. Ano siya, meron siyang more than one amino acid chain, kaya nakikita nyo yung dalawang magkaibang kulay na yan. Yung mga yan is nagre-represent doon sa amino acid chain. Tapos, they represent the highest level of organization in protein assembly and per definition, they are not held together by covalent forces. Secondary, tertiary, and quaternary structures of a protein are mainly stabilized by hydrophobic reactions and by hydrogen bonds. The contribution of hydrogen bonds to protein stability is strongly context dependent. Hydrogen bonds by side chains and peptide groups make similar contributions to protein stability.
The hydrogen bond also play a very important role in proteins structure because it stabilizes the secondary, tertiary, and quaternary structure of proteins, which form by alpha helix, beta sheets, turns, and loops. The hydrogen bond connected the amino acids between different polypeptide chains in protein structure. Hydrogen bonding confers rigidity to the protein structure and specificity to intermolecular interactions. During protein folding, the burial of hydrophobic side chains requires intramolecular hydrogen bonds to be formed between the main chain polar groups. The solubility, the solubility of protein in water is dependent on the ability to form hydrogen bonds with the protein surface. Proteins that have a greater hydrophilic surface content are generally more capable of forming hydrogen bonds with the surrounding water. The alteration of salt concentration of the solution is as is formed in salting out or in creates a shielding effect that reduces the ability to form an hydrogen bond with the hydrogens in water. The protein precipitation method of salting out utilizes this concept in protein fractionation.
Next naman is yung hydrogen bonds and carbohydrates. So first, carbohydrates are among the most abundant molecules in living nature, with the exception of water as monomers, oligomers, cyclic, and linear branched polymers. They occur alone or chemically combined with purines and pyrimidines in the nucleic acids with polypeptides in the glycoproteins and with lipids in the glycolipids. Carbohydrates are biological molecules made of carbon, hydrogen, and oxygen in a ratio of roughly one carbon atom to one water molecule. This composition gives carbohydrates their name. They are made up of carbo, which means carbon, plus hydrate, which means water. Carbohydrate carbohydrate chains come in different lengths and biologically important carbohydrates belong to three categories, which are monosaccharides, disaccharides, and polysaccharides. Next, like most biochemical compounds, carbohydrates are built of small repeating units or monomers, which form bonds with each other to make larger molecules called polymers. In the case of carbohydrates, the small repeating units are known as monosaccharides. So dito sa figure na to, yung parang kulay gray na anim dyan, carbon siya. Next.
Next slide.
I'm sorry, naglag. So, ito yung three categories ng carbohydrates, which is yung monosaccharides. Mono means one and saccharide means sugar. These are the simplest sugars. And the most common type of it is glucose. Pwede rin example dito is yung fructose and galactose. Next is disaccharide, which means two sugars. It is formed when two monosaccharides join together via a dehydration reaction, also known as condensation reaction of dehydration synthesis. And last is yung polysaccharide, which is a long chain of monosaccharide linked by glycosidic bonds. And ayun, ang example para sa disaccharide is sucrose, which is yung table sugar, lactose, milk. And sa polysaccharide naman, ang examples dito is starch, glycogen, cellulose, and so on.
So in hydrogen bonding in carbohydrates, the hydrogen bonds are of prime importance in stabilizing the conformation of molecules of carbohydrates and their fragments, in forming various structural formations, and in the process of orientation, crystallization, and ordering of supermolecular formations. Plants synthesize a structural polysaccharide called cellulose. Although cellulose is made up with glucose, the glycosidic linkages between glucose monomers are different from the bonds in glycogen and starch. This unique bond structure causes cellulose chains to form linear flat strands instead of coils. The flat cellulose strands are able to form tightly packed bundles. They are strong and rigid fibers result as hydrogen bonds from between polar hydroxyl groups in the bonded polymers. Tiny forces called hydrogen bonds hold the glucose molecules together, and the chains in the close proximity. Cellulose fibers provide structural support to plants. Next. So, our example of molecules or substance is chitin. Chitin is a large structural polysaccharide made from chains of modified glucose. Chitin is found in the exoskeletons of insects, the cell walls of fungi, and certain hard structures in invertebrates and fish. In terms of abundance, chitin is second to only cellulose. Basically, chitin provides protection and structural support for many living organisms, including forming the exoskeleton of shellfish and insects and the cell walls of fungi. So lastly, the importance of hydrogen bond in proteins and carbohydrates. Hydrogen bonds provide most of the directional interactions that underpin protein folding, protein structure, and molecular recognition. They satisfy the hydrogen bonding potential between main chain carbonyl oxygen and amide nitrogen buried in the hydrophobic core of the protein. The hydrogen bonds are prime importance in stabilizing the conformation of molecules of carbohydrates and their fragments, in forming various structural formation and in the process of orientation, crystallization and ordering of supermolecular formations. Together with carbohydrates, nucleic acids and lipids, proteins are fundamental building blocks of life. Proteins exhibit enormous diversity in their composition and hence function. Among other functions, they provide structure to a cell, allow movement, catalyze reactions, bring molecules across the cell membrane, and defend vertebrates against invaders. That's all. Thank you.